So before I start, I am slightly mortified to be on TV, as it were. They've given me a nice box I'm not supposed to leave. This morning, we're going to all pretend that this is my absent-minded professor Halloween costume, instead of what I put on this morning when I forgot I was giving this talk. With that said, the title, Applied Immune Profiling. This is something that never discount the value of luck in your career. I'm going to walk you through a series of happy events that have culminated in some very interesting things regarding perhaps the first time in my several-decade career that I've had an impact on the clinical care of at least one patient. So before I get rolling, declaration of potential conflicts of interest. The technology that I'm going to be describing has been commercialized. We patented it at the Hutch. We licensed it out to a company, Adaptive Biotechnology, that has been running with it ever since. I apologize for the fact that here in contrast to the Bay Area where I grew up, in Seattle, the profit motive is highly suspicious to all academics. But I will say that the interesting thing here was that when we came up with the technology, we did our darndest to get this sucker funded in terms of technology development grants in the academic space. And we were told either that it was trivial or it was impossible, but either way, they were not about to give us any money to do it. So we sought out some investors, did a little bit of it, and hopefully I can convince you that the technology, you can excuse us for the fact that we had to go outside of the NIH in order to make the technology work. So I'll start with a lucky lunch, and then we'll talk about T-cell receptor and B-cell receptor sequencing, and then some applications that have grown out of the technology that we developed. All of those of us who are in basic science have this dream of that one day that you sit down with the right couple of people. You didn't sit down with an intention to invent anything, and come away with a sketch on the back of a napkin. Well, it wasn't quite on the back of a napkin. It's on page 57 of booklet number 23 in my ongoing serial chronicling of my life in the lab. But I did sit down with the right two people, Hootie Warren, a clinical immunologist who treats a lot of leukemia and lymphoma at the Hutch, and Harlan Robbins, a computational biologist. And I have a long and deep experience in molecular biology. At the end of lunch, what began as a, hey, Chris, do you think that these new sequencers could be applied to T-cell receptors, had turned into a, we got to try this. And I will give you a little bit of foreshadowing. We knocked loose $5,000 in bid and proposal funding from the Hutch, which was absolutely fabulous. We went and we ran our first experiment, and a month after that lunch, we collected our first completely empty data set because Illumina forgot to tell me which strand they were washing off the chip. So our second experiment. At the time, there were a total of 10,000 T-cell receptor sequences in all public databases. Our second experiment, we had 10 million such sequences in one run. It's been a very cool ride ever since then. But I'm going to assume that I'm talking to a mixture of backgrounds. So adaptive immunity is all about that portion of your immune system that can't be hardwired. There are some things that just shouldn't be there. Double-stranded RNA, certain polysaccharides is what the innate immune system, which is all hip and cool these days, is all about. But back when I was in college, the adaptive immune system was cool. This notion that we have a basic problem on an evolutionary level, and that basic problem is that we intend to live for decades. If we don't live for at least a couple of decades, we're not even going to reproduce. In contrast, some of the pathogens that are adapting to us have life cycles where the generation time is on the order of 24 hours. This is a problem. They will adapt to anything that you are hardwired with, such that by the time that you are old enough to procreate, you'll be dead if you haven't got some other piece of you that can adapt as fast as these bugs. That's what the adaptive immune system is all about. You're not born with an antibody or a T-cell receptor against flu virus. You're given this really cool system of within the immune system, or starting from the hemocytoblast, we're getting down to the B and T cells. That's all we're going to talk about today. B does not stand for bone marrow, although they are derived from bone marrow. It stands for bursa fabricius, an organ that doesn't exist in humans. It's from birds, but that's where they first figured out what these things were. T, on the other hand, is quite convenient, the thymus. Anyway, so the B and T cells are what we're going to be talking about today, and what they do that's so cool, so very exciting, is instead of inheriting a receptor that's specific to every bug out there, what we are given is a chunk of the genome that operates somewhat like a card deck. Instead of having a specific receptor with specific exons that are going to be used, here I'm using the T-cell receptor beta locus as an example. There are V for variable region, D for diversity, and J for joining regions. And what happens is you've got 54 of those V segments, 2 D segments, 13 J segments. This is just for the T-cell receptor beta chain. And in the germline of the cells that are the cells that are becoming the T cells, as they develop. 330, just in case you aren't aware of this, is absolutely the low point of my consciousness on any given day. So we're going to have a couple of mind farts where there's a word that just isn't coming, and that was the first of them. Anyway, but instead of splicing is beautiful. Splicing, you get the exons in frame with one another 99.9% of the time. That RNA, you get the intron out of the way, and you're able to make the protein. This is sort of the opposite. This is brilliantly messy elegance in the sense that first, just selecting which V, which D, and which J, 54 times 2 times 13, you're up to a couple of thousand, well, 1,000. And that's great. This is like shuffling a card deck. But in addition to that, on the edges of these, where you join them, instead of having a nice clean splice for the exons, this is something where there's going to be a couple of nucleotides get nibbled away, so it might chew away a little bit from the D, a little bit from the J. And for those of you who are molecular biologists, which is probably none of you except for the ones that I actually recognize, terminal deoxynucleotide transferase, TDT, does not exist to put radioactive phosphates onto your oligonucleotides. TDT's job in biology is to insert random nucleotides at these junctions between the V and the D and the D and the J. So you're starting with which VDJ did you use? You combine that with a little bit of nibbling away. What reading frame is the D segment in? And now you insert a dozen random nucleotides, and you've gone to the point where there are more possible T-cell receptor betas than any one of us will ever make, no matter how many T cells we make in our body. There just aren't enough cells to get all the quadrillions of possibilities. Now, the beauty of these antigen receptors is that they act as heterodimers. So not only the beta, but the alpha in that T cell is also rearranging. So each T cell has just one of these rearrangements that's occurring that's going to be recognizing antigens. I won't get into the details for the immunologist. If you want to talk to me about bispecific and other fun things, we can talk about that later. But most of your T cells or B cells are capable of recognizing one antigen, and only one antigen. And the thing is that you don't know what you're looking for. It's a standing army of locks looking for a key. When a key shows up, then some subset of those locks, of those receptors, are stimulated. Those T cells will expand, they'll grow, they'll help you mount a cellular immune response. B cells, if they're stimulated, they're going to optimize their affinity, and then they're going to turn into plasma cells, home to the bone marrow, and pump out antibodies for the rest of their life. Really cool stuff. The biology of why it works is just amazing. Now, this was a black box. That is, even though the immune system had been studied, the number of these junctions, the specific sequences of antibodies or T-cell receptors that had been studied up until less than a decade ago was a few thousand across all of biology. So what we came up with was a core technology that, by using multiplex PCR, you land a forward primer in the V segments and a reverse primer in the J segments. Now, this is something that most people, when they're talking about multiplex PCR, they're talking about amplifying many different regions from the same set of templates, a bunch of different exons. This, in each cell, there's only one VDJ junction, or two, depending on whether both alleles rearrange. But which V and which J it is different between cells. So it's multiplex in the templates as well. And, well, Frankly, it had been done before, but nobody put them all in the same tube. Why did we put them all in the same tube? We wanted to put them onto a sequencing platform, one of these high-throughput Illumina, Celexa, the cool, sexy, hip, high-throughput sequencing stuff. Because this is the one chunk of your genome where we want to sequence it millions of times. The rest of the genome is not all that interesting after you sequence it the first 100 times. But this chunk is really, really cool. So that junction, this VNDNJ junction, is the complementarity determining region 3, CDR3. And for each T cell or B cell, in T cells, alpha and beta will rearrange the VDJ junction and in beta and the VJ junction in alpha. These are unique to that T cell. They're actually a perfect biomarker for following a subset of T cells or B cells because they are a sequence that is unique to that particular cell. So there is a downstream intron and C segment. That's all about making it operate. It's not about the specificity. And what we care about is generally, well, okay, the idea was let's go and sequence these things. Well, we did. And then we realized that if we're going to sequence something when we've got 50 forward primers and 13 reverse primers, we're going to run into some problems with bias. Which V segments amplify better than others? Which J segments amplify better than others? Well, this is a busy slide, so I'm going to skip through to the fun one. When we made we actually just synthesized all possible VJ combinations. And from all possible VJ combinations, we've now got templates that we've made where we know how much of it there is in the starting material. We throw it through the Multiplex PCR, and lo and behold, I don't know, there are just enough of you out there who look like you might be graduate students that I'm going to point out a few of the flaws in my slides. In this case, can anybody actually read my y-axis? Of course not. But trust me that that is a logarithmic axis, so there are some things that amplify very poorly, and there are some things that amplify very well, and it's at least 3 orders of magnitude in how well something amplified using our standard set of primers. This is a problem. It's a problem because we want to quantitatively pull things out. In a mixture of 10,000 different T-cell receptors, when we get the answer of 10,000 sequences, we'd like those to be quantitative rather than just qualitative. It was present or absent. Well, the first way you can sort of fix this is to measure your bias. Once you've measured your bias, then you can go back to the lab and you can start tinkering with the primers. Really crappy primers, just make a new primer. Really good primers, you can make them a little bit worse. You make them a nucleotide shorter or whatnot. After a little bit of tinkering, one can get them to a point where there's less bias. So now we're down to a range where it's probably about 1.5 logs of sequence of range. Now, it did turn out in our templates, since we made all possible VJ combinations, the bias was not a function of the combination. The bias for the V was strong, and the bias for the J was strong, but it didn't matter which V and J pair you were looking at. Because it's independent, that then allows us to say, okay, great. We now have something, and for those of you who may, I'm assuming some of you are MDs in the audience. MDs are notoriously good at math. So the important thing when you're trying to normalize a number, we're trying to get these to the true result, which should be a flat line there on the x-axis. To normalize things, the only important variable is that you know that there are no zeros. No matter how hard you try, you can't normalize 0 to give you a result that means something. If it was supposed to be there and you didn't see it, you can't fix it. But we now are seeing everything, and we're seeing everything at such a level that when we collect enough data, we're confident that we do see them. We know the bias from which V and J are amplifying, and we can then use computational normalization to take it down to the point where it's about plus or minus 1.5-fold for our precision of our estimates of the frequency of a template. In a mixture of millions of different templates, this is a big deal because we're very interested in the high-frequency templates and the low-frequency templates, and we want to be able to tell the difference between them with some precision. Now, I was all hot and excited about this technology. This was going to change the world because I wanted to do immunology. That is, when I first started working with Debbie Nickerson back in 1990-something, she sold me on, well, I wanted to be an immunologist. And she said, well, Chris, you can either go and study immunology, or you can just go back and do genetics and learn how to do the immunology of the future. Well, I'm convinced at this point that I've got the immunology of the future. We're ready to go. So we've started trying to do some work on type 1 diabetes and multiple sclerosis, and rapidly realized we had no idea what a normal immune system looked like. After about another 6 months of flailing to actually tease something out of what I thought we had invented this for, the low-hanging fruit kind of hit us upside the head. The heck with worrying about an immune response. There's times when you only care about one clone in a very complex background. So minimal residual disease in leukemias is a function of a clonal B cell or a clonal T cell that has expanded. This is your tumor. It can be a liquid tumor, it's a leukemia. If it's a solid tumor, it's most of the mass is somewhere, it's a lymphoma. Either way, it's derived from one B or T cell. And after you start treating a patient, there's a certain point where you can't see the tumor anymore. But does that mean it's gone? So coming up with a way to monitor this minimal residual disease is a big deal in the field, and it's something that has been done fairly effectively. The state of the art in this field was multiparameter flow cytometry, but we've now got a system where we can sequence and theoretically detect things at a frequency that's much lower than one might be able to get with multiparameter flow. When you're running a flow instrument, there's a combination of how much time you can spend on it and the false positive background rate. It's hard with multiparameter flow to get below a 1 in 10,000. Below 1 in 10,000, you're just plain in the noise. So, but you can see minimal residual disease down to 1 in 10,000 cells, which is pretty darn cool. Question is, could we do as well as or better than multiparameter flow? And what we're going to be doing is looking for these somatic rearrangements of the VDJ that are tagging a unique cell. The cell was a B cell or a T cell before it became a tumor, and if that rearrangement is still there, it's a unique sequence tag for the tumor. So, idea would be to collect CDR3 data from, you don't need many cells from the primary tumor. That is, when you've got the diagnostic specimen for ALL, some of these poor kids, the marrow was 95% tumor, 5% normal B cells, not hard to recognize what the dominant clone was. But here, that sequence, that junctional sequence, is a fingerprint. You can't go looking for a fingerprint without a reference that tells you who is the criminal. What is the fingerprint of the criminal? Because I can sequence and I can tell you I saw a million different junctions, but the junction of the tumor is not predictable. The junction of the tumor is a function of which cell was it that transformed. That's all it is. And so we need a diagnostic sample to start with to say, what is the fingerprint? Now I can go looking after I've treated the patient and ask, do I see that fingerprint again in many thousands or even millions of sequences? So when we go for the diagnostic specimen, now we're going into the post-treatment specimen. We're going to be looking for the same sequence to monitor minimal residual disease. Now this is the first sort of set of data that I found, and again, for those of you who have not taken the course How to Lie with Statistics, it's very important to recognize that the y-axis here is something kind of curious, that we don't have anything below that line. And that's because the minimum we could detect was 1 in a million, but we showed you down to 1 in 100 million. So you can ignore everything below that line, but the important point is here in blue is the frequency. This y-axis again is not, again, bad slide making, but I've done these things before and I just noticed it again. My y-axis is a log axis. So you've got things up here. This dotted line is 1 in 10,000 for detecting residual disease. 1 in 1,000, 1 in 100. We've got a couple of people where that minimal residual disease is not minimal by any stretch of the imagination. That's 5 to 10% of all their B cells are still the tumor. Those patients do not have a great prognosis for whatever therapy you've currently got them on. But the important point is that in blue we're showing you multiparameter flow, which sort of peters out at around 1 in 10,000. And then in red we're showing you for high-throughput sequencing, the same samples, same individuals, and we found almost twice as many individuals who had minimal residual disease, detectable tumor tags, and we actually found a few of them that were above this traditional 1 in 10,000 level that weren't picked up on by flow. So this is fun, exciting. So yes, we think we've got something that's more sensitive. Is it? Who cares? So it doesn't matter if you have residual disease below 1 in 10,000. So let's do a little bit more looking at the cool pieces. So is it relevant? Well, here, the Children Oncology Group has a number of different trials. I was lucky enough to start working with Steve Grubb, Mike Pulsifer on a clinical trial where they were looking at high-risk ALL patients doing autologous stem cell transplant. And they were looking at a treatment up front to see if we could improve survival and decrease relapse. Basic premise being wipe out or damage the immune system such that you can then replace it. So day 0 is when they do the transplant, and then they're either treating with tacrolimus or sirolimus afterwards, in the hope that you can improve patient outcomes. Now, for the clinical trial that was run, I had nothing to do with designing it. These are samples of convenience to me. And the sad part is that there really was no significant overall survival improvement. On a survival curve, you really want to see two things where they're diverging rapidly and staying diverged. And no, even with this different conditioning regimen, they weren't seeing a real improvement in patient outcomes. So that's the bad news. But here, if we look at day 0, at the time of transplant, you've gone through a conditioning regimen where we're wiping out the host marrow. Can we still see the tumor in day 0 specimens? So looking at MRD there, well, flow-based MRD does indeed predict relapse. So here, MRD negative, and I apologize, these charts I don't have the raw data for, so some of them are survival curves where they're top down. This one's a survival curve bottom up. If you're going to present such curves, generally one wants to stick with one direction being survival. But importantly, the difference between those who were MRD positive at 1 in 10,000 by flow and those who were MRD negative in green was quite significant. There was an improved outcome. And there was MRD positive at less than 0.1%, it seemed closer to negative results. But just being MRD negative didn't, there's still a quarter of them are going to have some sort of a relapse within the next, 3 years. Well, this is the standard of care. This is using flow and very competent flow people who I have tremendous respect for. So the question was, okay, in this same set of specimens, could we detect tags? That is, was there a fingerprint to be followed? So we went looking. And here, out of a series of, we had patients coming from T-cell leukemias, a mixed lineage lymphoma, a B-cell leukemia. Could we detect tags? Well, the number of patients here is 144. DNA was only available in about 2/3 of them. And of that 2/3, these 88 patients, at one locus, IGH, or on the second allele, or on TCR gamma or TCR beta, we were able to detect the fingerprint of the tumor, this unique rearrangement, in 84 out of 88, which was pretty good news. It's not perfect. We would have preferred perfect, but it is what it is. So now the question is, can we follow those tumor tags going forward? And in short, for the BALL, well, actually, sorry, this was for all of the patients in the trial. When you used a not 1 in 10,000, it was tumor present versus tumor absent by high-throughput sequencing. In this trial, we had some patients where we were detecting it closer to 1 in 1 million. It's only about 188 patients, so it's not enough patients to really tell the difference between 1 in a million, 1 in 100,000. But for this trial, we did have one relapse where they were tumor negative at day 0, and it was almost a 60% relapse if they were tumor positive. So at least to start with, we're doing better than flow because we've got a lower floor on our sensitivity. And these tumors they're fast-growing tumors. The fastest growing tumor available is Burkitt lymphoma. I'm not going to talk about that, but aggressive BALL is a very aggressive tumor, or can be. In this trial, it was. So it comes roaring back if you don't get it. And you can see that most of the relapses are within the first 2 years, after which time you've either cleared the tumor or you didn't. So in conclusion from this first trial, this was published last year, Mike Pulsipher is the first author, we can define unique tumor tags for 95% of the cases. We can detect MRD and any level, that is, many of them are below this traditional 1 in 10,000, did predict poor event-free survival. And detection of blasts, this is something that I didn't show the data for, but we also, if you detected them at any time in follow-up, that was also a bad deal. If they were MRD negative at day 0, but at day 29 you can see a little MRD, they were eventually likely to relapse. So closing the window on this piece of the story, this was a large collaborative effort with a large number of individuals, but Mike Pulsipher and Steve Grupp were absolutely critical. Now we're going to shift gears to a larger study that was a follow-up that I was not directly involved in, but is so relevant that I could not help but borrow their data and show it. This is 600 kids with ALL. So now we're asking, we've got enough numbers that we might be able to look at how well they're doing. And for those of you who are trainees, this is way too many words for one slide. Don't ever show this many words on a slide because your entire audience just started trying to read it. The only important things up here are we have a high-risk trial and a low-risk trial. They are about evenly split among the 600 patients. And we've got enough numbers that we can actually do some analysis on high risk and low risk independently. This is an even worse slide. And this is about how we defined the fingerprint of the tumor. I put it in there just to give you a notion for exactly how complicated the algorithm winds up being, but we were trying to come up with something that is an effective algorithm that you can sort of extrapolate across labs, across sites, such that you aren't just relying on technician X staring at the data and saying, I think there's something there versus not. This was an attempt to come up with something that was quite objective. So, using that complicated algorithm, when we look for that tumor tag, how many of these 619 patients could we see a tumor tag in the baseline active disease sample? Well, in that reference sample, we had 12 of them where the QC failed. So we're just going to toss out those 12, leaving us with 607. And of those 607, a majority of them had either an IgH or a T-cell receptor gamma rearrangement. That is, there were 39 that were only T-cell receptor gamma, there were 218 that were only IgH, and then there were 300 that had both of the above. Now, T-cell receptor gamma, this is a B-ALL trial. Why are we seeing T-cell receptor gamma? One of the joys of biology in terms of the adaptive immune system is that T-cell receptor gamma, for whatever reason, rearranges in virtually all adaptive lineages. That is, you can find T-cell receptor gamma rearrangements in alpha beta T cells, you can find them in B cells, you can find them in gamma delta T cells. It just is the first locus that rearranges when you start turning on RAG. So you conveniently can use T-cell receptor gamma as a tag for B-cell tumors. So, out of these tumors, we had 579, 95%, actually consistent with previous data. 95% of the time, we can see something. We've got a fingerprint for the culprit. The number, we had about 150 of these 400 where we had, oh, sorry, so 607. Now we've got post samples. These are, we're looking at, this was at day 29, end of induction. So after you have gone through full conditioning on this particular trial, can we still see the tumor? Or do we say that the patient is clear at the level that we could detect? Well, about a quarter of that 600 samples were not clear at 1 in 10,000. Another quarter were somewhere between 1 in 10,000, 1 in 100,000. And a third quarter were somewhere above 1 in a million. So there's fully half of the specimens are low frequency MRD, below the traditional threshold of 10 to the minus 4. So if you just use the 10 to the minus 4, the standard cutoff, the data clearly showed a significant difference between those who were MRD negative at 10 to the minus 4 and MRD positive using high-throughput sequencing. Was this a better result than we saw using multiparameter flow? The answer is no, it's actually exactly the same as the result that we saw using multiparameter flow. They're giving you the same basic answer. At 10 to the minus 4, it's either both are positive or both are negative. It's pretty infrequent that they're mixed. But there are a few. So here, using both high-throughput sequencing and flow positive, that's in green. High-throughput sequencing positive, flow negative, that's in red. And high-throughput sequencing and flow negative. That is, none of it could detect anything. That's in black. There were no high-throughput sequencing positive, flow positive, high-throughput sequencing negative samples. And here you can see that there is actually an improvement if you're double negative than if you're only negative by flow. So that's kind of good, but we're still seeing some relapse of patients or event-free survival. We're seeing events in patients who are negative by HTS. If you take the high-risk protocol, so we had two subsets of patients. In the high-risk protocol, these patients, you don't see a dramatic difference between any level, that is down to 1 in a million and below 1 in a million. But in the standard risk protocol, we saw a pretty striking difference, which was pretty exciting. That is, for your standard risk patients, if you get to the end of induction and we can't see the tumor at 1 in a million, this is a fantastic prognosis. The overall survival was similar, albeit the overall survival, we've got good rescue therapies, so the overall survival was not that bad even if you were MRD positive. Here was an interesting observation. If you're a B-cell tumor that doesn't have a B-cell marker, it has no IgH rearrangement, but it does have a T-cell rearrangement, is this a more primitive B cell, or is there something biologically different? Well, I can't tell you about the biology of how they came to be IgH negative, but for these 28 that were double negative and the 39 that were TCR gamma only, we did see that those patients where your germline IgH negative had a worse prognosis than those who were germline IgH positive. So these seem to be slightly more aggressive tumors. But rephrasing that, if you can't find a fingerprint in that reference sample, it's actually by itself kind of bad news, because those patients had a worse outcome than patients who did have a fingerprint, regardless of how much we saw in MRD. So, again, borrowed from colleagues, so overly wordy, but we did find that our technology, this high-throughput sequencing, is not inferior. I love the way that MDs speak about these things, but not inferior to the standard of care, multiparameter flow. It does capture additional patients, and it does have significant prognostic value in the patients if you're below the traditional threshold of 1 in 10,000. That is, these tumors are fast enough growing that they do have an increased risk of relapse, even if they're down to 1 in a million MRD. We did identify a subset of patients where the fingerprint's missing and they've got a worse prognosis. That's really kind of cool. At every decile that we look at, whether you're looking at 1 in 10 to the 4, 10 to the 5, 10 to the 6, the trend is for lower level MRD to have better event-free survival. In some subsets of the data, it's significant, but in all of the subsets that you can cut it on, there's a trend that goes the same direction. So in sum, we've got an accurate, more sensitive method than the state of the art for tracking MRD in these patients. Again, this was published just last year, Borowitz et al. And again, this work takes a tremendous number of individuals to do it and do it right. Now, I'm going to shift gears in the last 20 minutes and try and get back to my interest is, I started out with this, you know, we've invented a new microscope. I thought I was going to go look at immune problems with my microscope, not leukemias. Well, trying to bridge back to looking at the immune system is harder to sell to the NIH than one might imagine. But if you can connect it to something you've already successfully done, it's possible. So celiac disease is one of the basically, it's the bastard stepchild of autoimmune diseases. It's highly frequent, but because, oh, just don't eat wheat, there's an easy thing you can do about it. No one actually cares about treating it. For those of us who have celiacs in the family, we are very thrilled by the modern development of gluten-free foods, and the gluten-free bread is almost there. The gluten-free donut is not. But there is actually a reason you should still care about celiac disease, and that is that most celiacs don't know they're a celiac in the US, and of those who do know they're celiacs, they spent decades eating wheat. So they had a chronic stimulation of the largest lymphoid organ in the body, the gut, and in a subset of celiacs, even after they go on a gluten-free diet, they have residual risk for a very specific and very aggressive T-cell lymphoma, enteropathy-associated T-cell lymphoma, EATL. Now, this is kind of like you can smoke for a long time and you may not have cancer yet, but it's going to take a long time for your risk of lung cancer to drop back to non-smoker levels. Same sort of thing here. You take the stimulus of gluten away, you still have residual risk for decades after you go on a gluten-free diet for EATL. So how does this all work? Well, the normal gut is a highly oversimplified cartoon here, but it basically comes down to there's a layer of cells that are on the barrier to the lumen. Out here is the fecal stream. These cells are supposed to help you absorb stuff, and they're supposed to keep all those nasty little bugs that we have living in our gut out of the rest of you. So they have a very important barrier function. Well, just beneath that barrier, since these guys are not all that special, you've got a ton of T cells that are sitting there. You've also got B cells that can be resident, but the T cells are the real residents. And the gamma delta T cells in the gut are a large proportion of T cells, where gamma deltas are not a large proportion in circulation. And what's going on is your peptide, so here the gliadin is a little yellow triangle. Tissue transglutaminase on the way through the epithelial layer. Transglutaminase is the darn thing. And all of a sudden, for those of you who are unfortunate enough to carry DQ2, you've now got a presentable antigen that the antigen-presenting cells in the lamina propria do a pretty good job of presenting to CD4s. The CD4s turn on CD8s in the interepithelial layer, and presto, you have a grumbly tummy. Okay, you've eaten an antigen. This is an antigen that most of us is just another peptide that's coming out of the tryptic soup. But for those who are unfortunate enough to have tendencies towards celiac, they're now presenting it to their cells, and their cells think that gluten is important enough to start an immune response. Fortunately, if you take the gluten out, the immune response stops. This is one of the only autoimmune diseases we know of where you can turn it off just by stopping an exposure to an external variable. So the gluten-free diet, stop eating gluten, presto, stop having grumbly tummies. Unless you're the unlucky 5%. Well, actually, it's not 5%. So you come into the clinic, they think you might be a celiac. You either get scoped or you do a anti-TTG assay for antibodies, and we think you're a celiac. Okay, great. Let's take gluten out of your diet. 6 months later, you're still having the grumblies. Well, this is about a third of all patients, and most of that third just don't know how to recognize gluten, which is a really good argument for standard labeling of foods for whether or not they've got gluten in them. It is infuriating to have such random places on packaging in the US and then to go to another country and discover that there's a standard spot that you look for the G. Poof, I can't eat that. Okay, so enough editorializing. Among that third of GFD, gluten-free diet celiacs who are refractory, there's 5% of active celiacs won't resolve even if you do get the dietitian in and figure out that, oh yeah, that's also got gluten in it, okay? Which is troublesome because you have removed it. What's going on? Well, RCD1, it's really just defined as ongoing flattening of the villi that doesn't go away once you've gotten gluten out of the diet. But RCD2, refractory celiac 2, you start to see aberrant intraepithelial lymphocytes. That is, you're seeing these, what should have been CD8 lymphocytes in the IEL layer, but now they're CD8 negative. And they stop expressing the T-cell receptor on their surfaces. This is a pre-malignant condition. And of the RCD2 patients, so about 5% of RCD1 become RCD2. Once you're RCD2, within the next decade, it's something like 5% risk of developing enteropathy-associated T-cell lymphoma. Now, this being a lymphoma that is deep in the gut, it is very rarely detected early. It is a kind of the ovarian cancer. It's in the middle of you. You don't notice it until you've got symptoms of some sort. And by the time it's noticed, this is a very aggressive T-cell lymphoma. It's hard to treat. So trying to prevent this would be a fun thing to do. Well, so that was where I was interested in going there. Now, what we do know is if you don't have HLA-DQ2 or the rarer DQ8, these are the two HLA types that are presenting the gliadin molecule on the surface of the cells. If you don't have those two, you don't have celiac. Basically, it's an all or nothing, which means that for an awful lot of people who are absolutely certain that they are gluten sensitive, if you're gluten sensitive and you're not DQ2 or DQ8, thank you for contributing to bringing the cost of gluten-free food down for the rest of us who actually need it. but it really doesn't do any good for you. It just basically is not a big deal. But anyway, so the DQ2 is presenting it. CD4 that recognize DQ2 are expanding. These epithelial CD8 are not necessarily recognizing gluten directly. So the IELs might not be derived from the original trauma. So we wanted to look at, within an individual, how oligoclonal was this T-cell response? So looking at active celiacs at a time point where we've got their pre-gluten-free diet, and then we look at them after they assume a gluten-free diet. This was the model that we were going with. It turns out it's kind of hard to get multiple samples from people who no longer have a grumbly tummy. So we wound up working with a set of samples where some of them were, we did have Epithelia and lamina propria, so the two different layers. From paired, we had 7 active celiacs who hadn't gone on GFD yet. We had 5 gluten-free celiacs who'd been on it for a while. We had 2 refractory celiac 1, and we had 6 normal controls. RCD2 is rare enough that we were just trying to get all the cylinders firing on this project, so we didn't use the RCD2 samples yet. And we collected both Tcell receptor beta because that's the CD4s, we're pretty certain of it, but we also collected gamma because gammas are such a large proportion of the intraepithelial lymphocytes. Well, if you look at the intraepithelial lymphocytes versus the lamina propria lymphocytes for T-cell receptor beta, here's just one person. X and Y axis are IEL versus LPL, and we're plotting on a log scale the frequency of any given clone. What you can see is that there are high frequency clones are observed in both. By the time you get down to the low frequency stuff, it may or may not be observed. Translation, it's harder to get the epithelia cleanly separated from the LPL. They're not really that distinct as compartments of T cells. Oops, sorry, I just stepped outside of the magic box that they put me in. But this is if we're looking at the same person. Now let's look at the IEL versus LPL from 2 different people. There is nothing, there is no public T-cell receptor observed between 2 individuals, even though we're talking about potentially exactly the same condition, 2 active celiacs. So the T-cell receptors really are, in this case, they're not necessarily snowflakes, but we certainly didn't see anything that was a uniform, everybody's making a public T-cell receptor. That was a little bit frustrating, but where it got interesting, and this is something that's been reported. T-cell receptor stereotypes have been reported by pulling them out of celiacs. So which gliadin fragments, if you have the right fragment, you have the HLA molecule, you have the gliadin fragment, you can actually go fishing and find T-cells that recognize this particular combination. And it turns out that when one digests the gliadin, there are a whole bunch of different fragments, but alpha-2 here is immunodominant. Across individuals, that's the one that's showing the most clones that are recognizing it. So you can then make a tetramer for DQ2 paired with gamma, gliadin alpha-2. Take that tetramer and go fishing for T-cell receptor, for T-cells. T-cells that light up, you sort them by flow and you start sequencing. Well, this was done by a group in Norway who are extraordinarily good at making tetramers. Tetramers are an art form. And they found stereotyped responses. That is, here there was a particular length of a CDR3 where in red is a particular amino acid that seemed to matter. And you could see several different stereotyped things, but not bothering to get into the details. I'm just showing this to you so you can see that there's a column that is red. There is something there, and the biggest sequence, this, I'm not being crude, alanine, serine, serine something, arginine, okay? But assortidity is the consensus that they were seeing across T cells that were pulled out by this particular tetramer, okay? Which was really pretty cool, and so they Chow et al. Reported not half of their celiacs had this particular tetramer positive clone, which was really cool. My problem was in our samples, Zippo. Not Zippo, sorry, they're normals. I mean, 9 out of 17, 0 out of 6. 0 out of 6 is a good number. So there's something interesting there. But when we go and look at it in our samples, we didn't see it. Oh, sorry. It's been too long. I plead 330. This is my data. This isn't their data. This is in our celiacs. We did see half of them were positive for it. But the frequency that we were seeing it at is not 1/4%. It's 1/4%. This is not a dominant thing that you would have said, aha, I can see an immune response. This is something that's way down in the background. There are hundreds of clones that are more frequent, but this one is seen in half of our celiacs, so there is something there. It's just that the stereotypy of the response is not particularly high frequency for the stereotype clones that do get pulled out. But gamma is where things got interesting. So when we looked at, here again, it's looking at within an individual versus across individuals. The gamma sequences, there is gamma overlap between individuals. There are clones where they are sequence identical between individuals. And one can ask, okay, well, for these two individuals, we've got a certain number of clones that are in common, and those clones, some of them are high frequency, some of them are low frequency. All of us will have a tendency to go back to our roots. I'm a population geneticist. So when I'm comparing two different populations, I want to see how different, how far are they from each other. There are great statistics for this. I just borrowed Ney's genetic distance to ask how similar is the repertoire of gamma between any two individuals and built a tree off of it. Well, that tree actually, I'm not going to show you the pictures because we're short on time. There's the tree. What we found was here, distance, the longer the branch, the more different a pair of samples are. Since we had epithelial and lamina propria from individuals, you can see most of these, you have two samples that are from the same person down here at the bottom, and they're various distances. Now over here, not highlighted at all, are 6 samples that we had from blood. In blood, there is one dominant T-cell receptor gamma sequence that's seen in all humans. It's recognizing something. I can't remember off the top of my head what it is, but it's actually known what it's recognizing. And that particular rearrangement is something like 10% of your circulating T-cell gammas, which makes, because all of them have 10%, and there is blood in our sample here, those blood samples all clustered very tightly. They've got a very common thing shared between all of them. But what was more interesting was that here, this may be bad for those who are color challenged. I sometimes plead color challenged when I get dressed. I have no actual biological excuse. But the celiacs versus the normals were further apart from one another than the celiacs were. There's something here on the tree that the y-axis is the only dimension that matters, and the tree distance between any pair of purple and red was shorter than the distance between these blue normal controls. So something is going on in terms of the repertoire. And without reading my slide to you, I'll summarize it as we didn't find a stereotype in the T-cell receptor beta, but here, when you start lining up the clones by frequency and asking what are clones that are shared by our celiacs and not by our normals, there are some standout clones. This particular sequence, Catwood or Peckliff, is my favorite because it was less than 1 in 100,000 of the T-cell receptor gamma sequences for any of our 6 controls, and our 6 controls did include DQ2 controls. So this isn't just something that's coming up in someone who's DQ2. You have to be DQ2 and a celiac. But more interesting was it was in 13 out of 14 of our celiacs. What about number 14? Touchdown. Number 14 is the DQ8 celiac. Different HLA type, probably not presenting exactly the same things. This is a public T-cell receptor gamma sequence that is somehow involved in the celiac disease process. We're currently trying to replicate this. We haven't figured out exactly what it does yet, but it was, you know, when you've got, it's not 13 out of 14, it's 13 out of 13 DQ2 celiacs versus 0 out of 6 normal controls. That doesn't require complicated statistics to say it's interesting. What it does require is a replication sample, which we're currently working on. And in the interest of time, I'm going to blitz past these guys, except to say not only was it shared between individuals, but when you looked at Catwood or Peckliff, in the K codon, in the lysine, in the lysine codon, the last position is a wobble position. And we would see Catwood or Peckliff in some patients. That is, the same patient, individual 1, has A, C, G, and T clones. This person has made that Catwood or Peckliff thing at least 4 times independently in different T cells, which is kind of cool. It's not just recurrent between individuals, but within individuals. So the working hypothesis at this point is that this is, you know, historically 1%, and this clone turns out to be 1% of the gamma sequences in most of the individuals. That's a lot of cells when you're extrapolating it out across the whole gut. So we're currently chasing this guy down, and what's interesting is that it does show up not only in active celiacs, it persists in gluten-free, and it persists in the refractory celiacs. So we think that this might be something that gives us a handle on, did it start, is this just a bystander to the disease process? It's just there. Well, we can rule that out because it wasn't there in any of the controls who were DQ2 positive. But we can't rule out whether it's a rioter or the criminal. Did it start the disease process, or is it just something that gets amplified during the disease process? We don't know that yet. So we're having a fun time chasing this, trying to figure out, is this stereotype recognizing DQ2 something presented by DQ2? And gamma delta is actually, the current model is, it's actually HLA independent. So what the heck is it recognizing? But it's got something to do with the disease process, and all of that could not have been achieved without Govind Bhagat, who's a fantastic collaborator at Columbia, and Ludwig Solid, who's the master of all things Tcell, receptor, tetrad in gliadin in Norway. So just summarizing the overall things I've exposed you to, I apologize for the deceptive advertising. In my title that was circulated, I had the word immunotherapy. There are cool applications of this technology in looking at tumor-infiltrating lymphocytes and the diversity of those lymphocytes. That's not actually my work, and when I finally put my slides together and realized there's no way I could fit that into a 1-hour talk, so you'll have to get someone else out to talk about TILs, but immunotherapy is the first place we're getting polyclonal stuff to show interesting results from sequencing like this. So our applications, minimal residual disease is absolutely working. It's now actually clinically available. The clonality of tumor-infiltrating lymphocytes is something that is beginning. Screening for stereotyped receptors, this is just my personal interest. This isn't something that is being provided as a service. And then profiling the immune responses is something, I think we're finally starting to turn the corner where we can go back and take this microscope and look at the immune system, whether it's in pathogens or autoimmunity. Thank you very much. Have time for a few questions, but you're also welcome to duck out if you got to make it to the 4:30.
Well, I'll start with a question. Yeah. So my question, it has to do with minimal residual disease.
Yeah.
And using the test to maybe guide whether your conditioning therapy has reached the point where you should stop or you should continue. Is that, looks like you're doing that.
Well, that's something where it's so frustrating how slow it is to design clinical trials. Because for chemo, for many conditioning therapies, and sorry, I'm about to embarrass myself, the difference between conditioning and other chemo, but if you've got 6 cycles, have you got a patient who's clear after 5 cycles? Or at the end of 6 cycles, are they not clear? It absolutely does make sense, but the clinical trials are going to take years to get them going to prove that, yes, you can make a clinical decision. For those of you who don't speak chemotherapy is just, all chemotherapies are poisons. And the key is poisoning the tumor faster than you poison the patient. This is where there's a big advantage in being a child taking chemo. Kids can take doses that adults can't. But, you know, have you successfully poisoned enough of the tumor? Getting back to the population genetics side, the goal is to drive the tumor extinct. Getting the tumor down to 0 is what you're after. And if you've achieved that in 4 cycles, maybe you don't need to continue with additional cycles. Or after 6, maybe you need to do 7. So, but no, we don't know the answer to that yet. It's a good question.
Anybody else have any other questions?
Yeah.
Actually, I have 2 questions. The first question is, how much does this cost, this test, which is clinically available? And secondly, what is the recommendation in terms of, in the setting of MRD, you know, you showed that it was non-inferior to flow. So is it to, you know, you do flow and then you do, you know, and the cost, of course, is probably a factor in.
Right. And this is something, those are very good questions. And the sad part is I'm actually not qualified to answer them because I'm the nerd doing the lab side, not the people doing the clinical side. It didn't take me very long. In my introduction, he didn't mention the disastrous year I spent in industry, discovering I don't belong in industry. When it comes to something, the clinical test is available. I don't know how much it costs clinically. I know how much it costs on a research basis, but I don't know what the clinically validated and basically all the CLIA certified stuff actually costs. It is available, and I'd have to refer you to folks at Adaptive to give you the answer to that. Yeah, let's be honest, it probably is. But I don't know what the actual number is. As far as deciding on using this versus using multiparameter flow, it's something where in the long run I can see this supplanting multiparameter flow because we've got additional studies coming through the pipe suggesting, consistent with what I showed, that a threshold below 10 to the minus 4 actually is still clinically relevant, and you can't get there using multiparameter flow. The other advantage is we're just working with DNA, so the sample can be a bit more abused than if you have to do flow. You need cells that are intact. But multiparameter flow is in a lot of different places already. So it's not something where you immediately have to switch horses, but it's definitely something to keep it on your radar because I think that it is going to have increasing numbers of studies suggesting not just non-inferiority, but superiority in certain applications.